Welcome to Lecture 18 on translation. So when we show proteins, different than when we show DNA and RNA, we show basically these large blobby structures. But what's important to remember is that those blobby structures are in fact long chains of amino acids. All right. The reason they have those blobby structures is because the long chain of amino acid, which is encoded for a nucleic acid sequence, forms a three-dimensional structure that folds up on itself. And it's the folding of the three-dimensional structure that allows proteins to do what they do. It allows them to be enzymes. It allows them to function in metabolism. It allows them to form critical structures. So the folding aspect of proteins is an extremely important aspect of biology. So just to remind you, just continuing on this point, that proteins are long chains of amino acids. Here we have a DNA sequence, okay, which encodes for an mRNA, and after translation, it will code for a protein, all right? But mutations in the DNA sequence will get carried informationally into the mRNA sequence, which would then be translated in the same positions, all right, corresponding to the nucleotide sequence of the mutations from one amino acid to the other. Okay, so the positions of mutations in the genetic map are the same linear order as amino acid replacements in mutant proteins. Now, in the same way that nucleic acids have a 5' to 3' polarity, so do proteins. In fact, so do amino acids individually. Amino acids are basically comprised of this particular structure. There's an amino group on one end, right? There's a carboxyl group on another end, right? And then there's a critical side chain which actually determines what type of amino acid a particular amino acid is. All right, but it's the amino group on one end and the carboxyl group on the other end that establishes a form of polarity in the long chain of amino acids. So in this particular case, you have a side group R1 and a side group R2 and a side group R3, and this forms a sort of set of amino acids, and it's the carboxyl end reacting on one amino acid with the amino end of the other amino acid that links the amino acids together through something called dehydration synthesis. So at the carboxyl end you have an OH, at the amino end you have an H. Those two are combined to form a water molecule, right? And a peptide bond is formed. So here you form a water molecule, the water molecule goes away. Here you form a water molecule, it goes away, and a chain of these three amino acids are formed through these two different peptide bonds. So what you'll end up then in the primary structure, you'll have an amino end on one end, and then you'll have a chain of these amino acids, all determined by the side chain, all right, this R1 or R2, all right, and then terminating with a carboxyl end. And this is the primary structure, it's just the linear sequence of amino acids. But as I said earlier, is that that's not what determines the functionality of a protein. What determines the functionality of a protein is how the primary structure adopts a three-dimensional configuration within the cell. And then adopts that three-dimensional configuration through the secondary structure. Right? And so there are different ways in which hydrogen bonds between amino acids at different positions in the peptide chain can interact with one another. And so you can end up with helices, known as the alpha helix here, based on these forms of hydrogen bonding between all these different amino acids. You get structures that are known as pleated sheets. Here we have hydrogen bonding from these different amino acids at different lengths in the chain. And then once you have a secondary structure, that secondary structure in complex with other molecules, here we have a heme molecule, but you can also have zinc, you can have all sorts of other critical ions, can form a tertiary structure which combines the amino acid sequence with these other molecules. and then those tertiary structures can combine with other proteins. Here, for example, in hemoglobin, we have the alpha hemoglobin and the beta hemoglobins, can form a larger molecular complex. And one of those complexes we've been talking a fair bit about is the RNA polymerase complex, which contains multiple proteins. So, right after the structure of DNA was determined by Watson and Crick with the work of Rosalind Franklin, right after that was found that the DNA sequence coded presumably amino acids, it was very fast to realize that the code needed to be decoded. It had to be cracked. And so at the very beginning, it wasn't sure if it was like a two-way code where each pair of nucleotides coded for amino acid. It was hard to figure out how that would work, because then there would be 16 combinations of 2, and those 16 combinations would be difficult to understand how they might encode for 20 amino acids, because biochemists, of course, knew that there were 20 amino acids. So, then you could say, well, maybe each of the 2, and then there's some overlap, or, you can imagine all sorts of different ways in which DNA sequence could code for protein sequence. But it turns out that it's a 3-way code. There's a 3 nucleotides per each codon. And this was determined originally with sort of a lot of different what are called in vitro translation experiments, where one can figure out, if you can generate a translation machinery that you can put in a test tube, and then you can put RNA into the test tube, and then see what proteins come out, then you can start making inferences about the way the code is structured. So, for example, if you just dump an RNA sequence into your test tube that has this translation machinery, it has the ribosome in there, it has a bunch of U's in it, a bunch of uracils, and you spit out phenylalanine, well, okay, then you know that a bunch of U's, here we have the codon table, encodes phenylalanine, but that doesn't necessarily tell you whether it's a 1 or a 2 or a 3 nucleotide code. It doesn't tell you that. And so you can also put in maybe a repeating sequence of AGA, 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 and you can find out, you can look on here, and it's right there. It encodes for arginine. So it was a lot of, trial and error of, putting different sort of chains of nucleic acids into these tubes and figuring out what came out. Eventually, it needed to move past that, and I won't get any further into the detail of how this was all figured out, because it's remarkable. But if you're interested in the story, I really, really recommend this book, Life's Greatest Secret by Matthew Cobb, because it really explains how this whole thing went down. It was a great book. Okay, so the translation itself, of course, is performed by the ribosome. Just a little bit of a reminder, here's a prokaryote with a circular DNA molecule, the mRNA being produced, and then translation occurring right on the mRNA as it's being transcribed. But of course, the ribosome in eukaryotes resides in the cytoplasm, so the RNA molecules that are translated are fully transcribed at the time that they're translated, because they have to have left the nucleus. So this is a 3-dimensional structure of this very complicated molecular machine, and it's a structure that comprises both proteins called ribosomal proteins, but it also comprises important RNA sequences known as ribosomal RNAs. All right, so it's a mixture of RNAs and proteins. And actually, it's an interesting thing that the fact that the ribosome consists in part of RNA itself, because RNA is a fascinating molecule. RNA, like proteins, can fold in different ways. And actually, there are RNAs that can actually have enzymatic capacity. But the fascinating thing about this molecular machine, which consists of RNA and protein, is people think it's a hint that before there was a world of protein, perhaps there was a world of just RNA. Maybe DNA, but there's this thing called the RNA world hypothesis, and it's been proposed that the earliest replicating molecules were RNA, because they have the ability to encode information in the nucleic acid sequence, but they also have the ability to form functional properties through the way that they fold. All right, but anyway, so the ribosome is this large complex of proteins and RNAs, and the ribosome can be broken up into what are called subunits. So the large 80S ribosome can be broken into a 60S ribosomal subunit and a 40S ribosomal subunit. And the 60S ribosomal subunit will consist of what is called the 28S ribosomal RNA, the 5.8S ribosomal RNA, the 5S ribosomal RNA, and 49 proteins. And the 40S subunit consists of 18S ribosomal RNA and 33 proteins. The way that translation fundamentally occurs, though, there has to be a bridge between the world of nucleic acids and the world of amino acids. And this is mediated by transfer RNA. Transfer RNAs are the bridge between these two forms of information content. Okay, here's a structure of a tRNA molecule. It has this sort of hairpin kind of funky structure. But the critical part of this is what is called the anticodon loop, and it's the anticodon loop that recognizes the particular stretch of sequences within the codons of the RNA. Here you can see that it's a reverse complementary, C to G, G to C, and U to A here. And this is how it might look like a little bit more, looking at it from a more structural perspective as opposed to a cartoon type of perspective. Now, so they had their bridge between nucleic acids and amino acids, and the way that that occurs is that they're, quote, charged with an amino acid by an amino acid tRNA synthetase. All right, and so each of the different tRNAs has a particular amino acid tRNA synthetase that it can bind to, and there's a little pocket inside this protein. The protein is the blue. I'm not showing it as a three-dimensional crystal structure, but it's a sort of a cartoon. There's a little pocket that binds, in this particular case, the tRNA for alanine, CGU, and then there's also a pocket for the amino acid. Right, so there's the alanine there. So these two pieces of this process are bound inside this amino acyl tRNA synthetase, and using ATP, these are joined. The amino acids are coupled to the 3' end of the tRNA. Now, one thing that's interesting is that we have noted that there's redundancy, of course, within the codon table. All right, because there are 20 amino acids, all right, but there are 64 possible codons, so there has to be redundancy of sort, and the source of that redundancy can be seen here, because some tRNAs have this sort of lack of specificity into the codons in which they bind to. So here's a particular tRNA codon, AGC, which recognizes UCC, but notice here it also recognizes UCU. So this position here is known as the wobble position, and there is some lack of specificity and some tolerance to mispairing here. And because there's some tolerance to mispairing at this particular position, that means it's the third nucleotide that tends to be the nucleotide that's the least critical for determining what amino acid is coded for by a particular codon. Okay, so with that perspective in mind, there's an mRNA sequence and it has to be translated, all right? And the way that this occurs is going to be different between prokaryotes and eukaryotes. A critical difference between prokaryotes and eukaryotes, there are many, but one of them is the fact that prokaryotes do not have that cap sequence at the 5' end. What that means is that translation is initiated when the mRNA is incorporated into the ribosomal RNA, and there's a base pairing between the ribosomal RNA inside the ribosome, okay, and a sequence called the Shine-Dalgarno sequence. Okay, so there's an mRNA molecule that comes into contact with the 16S RNA within the ribosome, and there's this base pairing that can occur. And once that base pairing is identified between a particular mRNA sequence and the 16S ribosomal RNA, a start codon that's downstream will initiate translation. But eukaryotes is different, right? Because the cap plays a critical role in basically bringing an mRNA molecule, all right, to the 40S subunit. All right, so the 40S subunit will have a methionine tRNA embedded within it, and will recognize the cap. All right, and that methionine tRNA can kind of basically scan along the length as the 40S subunit moves along the length of the mRNA through the recognition of the cap sequence. All right, so it moves down, moves down, moves down, and here it recognizes a AUG sequence. All right, the tRNA recognizes that, and the tRNA, of course, is coupled with the methionine. And once that recognition occurs, the next subunit, the 60S subunit, comes in, which completes the formation of the 3 sites where the tRNAs will come in, form the peptide bond, and then exit. All right. So sort of another way of showing this, there are these factors called elongation factors. There's initiator tRNA, and then there's this cap sequence. And the elongation factors can be bound to that cap sequence. And then in this particular complex here, the elongation factors will allow for the 40S subunit to bind to the cap and then begin scanning along. Now, once the scanning along occurs, these initiating factors fall off once that methionine recognizes that first subunit, and then the 60S subunit will come in and join once these elongation factors basically are lost. Okay, and this leads me to the introduction of the 60S ribosomal subunit, which is where the tRNAs will come in, form the peptide bond, and then exit. And they are given designations called the aminoacyl site. And it's called the aminoacyl site because it recognizes the tRNA. And then there's the peptidyl site, which is playing a role in the formation of the peptide bond, and then the exit site. So here we have the very beginning. The 40S subunit is and the 60S subunit are coupled now. The methionine tRNA is recognizing the start codon, and at this particular point now you have 2 tRNAs. You have the second tRNA has entered the A site, or the aminoacyl site, and the peptide bond is then formed between, in this particular case, methionine and phenylalanine. All right. So then this will then further process down the length of the mRNA. Kind of toggle down, and now they'll move their positions within the ribosome, and the A site will then be emptied, the P site will be filled, and then the exit site will be filled with the methionine tRNA that no longer has the methionine actually coupled to it, and the methionine and phenylalanine will have formed a peptide bond. So now the A site is empty, the A site fills in, okay, and then another peptide bond is then formed between phenylalanine and this amino acid, okay? And then the A site will be empty again, and then that methionine tRNA, which is no longer carrying the methionine, will be ejected from the E site. All right, so it's a sort of flow of tRNAs from the A site to the P site to the E site, and during that process, these tRNAs allow for the sort of tethering of these amino acids together. When it comes to stopping, though, all right, it's important to remember that while there is a stop codon, all right, there is no stop, amino acid, and there's no stop tRNA. All right, so what we have here is a stop codon now enters the A site, and instead of a tRNA coming in here, because the tRNA doesn't recognize that particular codon, another factor called the release factor. So in this particular case, it's called RF1 comes in. And once the release factor enters the A site, then the ribosome, the two subunits come apart, the tRNAs are released, and then the long amino acid chain will also be released. So I want to ask a quick question because I would like to bring this all together. In the DNA sequence, the order of regulatory sequence goes. So we're talking about a gene here, and I want you to think about which one of these is correct, all right, in terms of the order within the gene, all right? So I'll take a moment. Okay, in the DNA sequence, the best answer is A. Transcription start. In the DNA sequence, that's where the mRNA transcription begins. All right. Then the mRNA is transcribed, and at some point in that transcript, all right, a nucleotide sequence, a 3 base pair nucleotide sequence, will be placed in the mRNA, and that will be the translation start codon, right? The one that codes for methionine. All right. Now that nucleotide sequence will be embedded in there, in that mRNA molecule, but translation, of course, won't happen yet. All right. And then the mRNA will continue to be transcribed, and the translation stop sequence will be transcribed inside that mRNA molecule. And then finally, from that DNA sequence, there'll be the transcription stop. And that's the sequence that tells the transcription to end. So the translation start and the translation stop are embedded in the DNA sequence within the transcription start and the transcription stop. So just as one last point, yeah, sort of saying what I just said before, here is the RNA molecule, right, which got transcribed from a DNA sequence. The RNA molecule originated here at the transcription start in the DNA sequence. It ran all the way along, synthesizing off of the DNA template, and then terminated at this particular transcription stop. But within that mRNA molecule, at one end is going to be the translation start at the 5' end, and the 3' end, the translation stop will occur. And again, those are embedded within the RNA sequence, coming from being embedded in the DNA sequence. And at the beginning of this, you'll have a 5' UTR because this RNA isn't translated until this is encountered within the ribosome. And then translation occurs, and then the stop codon is confronted, all right? And then this RNA molecule will be released from the ribosome, but there will still be remaining RNA that does not get translated.